My name is Ana Isabel Aranda, and then I'll be comparing the AKT gene in both lung and prostate cancer pathways. If AKT plays a role in inhibiting apoptosis, then it may be a key factor in cancer-related pathways because its regulation of cell survival could influence tumor progression. So I decided to look at prostate cancer and non-small cell lung cancer or lung cancer to see what they both had in common, and I was able to gather my networks through Index, analyze them through Cytoscape, and compare both of their genes through Benny. I observed the heterogeneity, degree of centralization, properties of the networks, and other statistical numbers, and came down to the conclusion that AKT does indeed play a role on cell survival in cancer pathways. So according to the National Library of Medicine for both cancers, for the non-small cell lung cancer, it's the most common type of lung cancer, accounting for 85% of cases worldwide, and is characterized by abnormal cell growth in the lungs, often linked to smoking, environmental factors, and genetic mutations. Key pathways are AKT pathway and the EGFR signaling. Prostate cancer, on the other hand, is common in men worldwide. It arises from abnormal growth in the cells as well in the prostate gland, and it's associated with age, hormonal factors, and specific genetic mutations. Important pathways here are also AKT signaling and androgen receptor pathways. The AKT gene, part of the PI3K, AKT, and mTOR signaling pathway, helps regulate important cellular processes like growth, metabolism, and survival. It inhibits apoptosis to make sure that the cell survives under stress, and it controls the cell cycle for proper growth and division. However, in cancer, AKT is often mutated or overactivated, leading to uncontrolled cell growth and resistance to cell death. And in lung cancer, the activation of AKT is linked to mutations in upstream regulators like the PIK3CA or loss of tumor suppression like PTEN. In prostate cancer, dysregulated androgen receptor signaling activates the AKT, which enhances the effects of the AKT pathway and promotes tumor progression. So understanding cancer pathways is important, and systems biology helps by integrating complex data into networks that reveal the gene, protein, and the different pathway interactions. Since cancer involves multiple pathways, studying these networks can help pinpoint the key players like the AKT gene, and will help approach understanding of how the pathways influence the tumor behavior. I was able to get this AKT information on an article that was targeting the kinase AKT mTOR signaling pathway. So first, I obtained the pathways from the MDEX website, and then since I'm working on a related project outside of this class, I chose to analyze the cancer-associated networks, specifically the lung cancer and prostate cancers, which I loaded onto Cytoscape to help me visualize and analyze with their tools and look at properties such as clustering, coefficient, neighbors, etc. Then I made a clear graphical representation of the networks for both cancers, which included the nodes, edges, and interactions. To further compare the two networks, I extracted their gene lists and then used Vinny to make a Venn diagram, which helped me look at the several genes that both networks shared, such as AKT, PDPK1, and TP53. AKT stood out most to me because it was the most appropriate gene for further analysis since it had a hub in the networks, and it also caught my attention because of its direct connection to the survival node. So once I obtained the network statistics, I compared the analysis of both pathways, and then I generated this image in Cytoscape where I can see the AKT gene and the neighboring nodes. In this graph, each node represents a gene, protein, or phenotype, and then the edges between the nodes indicate the interactions, either activation or inhibition. Despite both networks having fewer than 22 nodes, the hubs around key genes are very noticeable. For example, the prostate shows a clear hub around AKT1, but in the lung cancer, there's not such a clear hub around AKT, but there definitely is a major hub around PI3K. So AKT is more of a secondary role here. It also shows the common nodes, such as the PDPK1, which regulates AKT in both pathways. It's also important to look at how the AKT interacts with survival, proliferation, and metastasis nodes in the prostate cancer, while in the lung cancer, there's no metastasis node, and the pathway to proliferation is much longer. So, to understand the networks in greater detail, I looked at several statistical properties, such as the degree. So AKT interacts directly with 11 nodes, making it a significant hub. This includes both in and out degrees, showing that the AKT regulates many other genes and is also regulated by genes. Additionally, in the lung cancer, we can see that the AKT only has 3 neighbors, way fewer than the prostate cancer. But despite this, AKT is still a key player since it regulates survival and cell death inhibition. It's more of a secondary hub here. The primary hub would be the PI3K with a degree of 5, basically the highest in this network. So both pathways have AKT nodes that directly connect to survival nodes, as well as PDPK1 nodes feeding into AKT. However, prostate obviously has more nodes, such as the metastasis and straight direct edge to proliferation, not like the lung cancer. We can also see that both networks are relatively small and sparse, with an average of 2 to 3 neighbors per node, compared to more dense biological networks. This sparsity suggests minimal and targeted interactions, which aligns with the idea of a small world network, where nodes can still communicate efficiently despite having fewer connections. Also wanted to note the heterogeneity. So in prostate, it's 0.744, which suggests a greater likelihood of dominant hubs, such as the AKT, indicative of a scale-free network structure, where a few nodes hold many connections. But in the lung cancer, there's a lower heterogeneity, meaning it's a more distributed network with some clustering and shorter path lengths. It is not likely to be a scale-free network. Both networks do have low clustering coefficients, less than 0.1 for both, and since it's so low, basically means it's tightly connected groups or very close-knit communities, explaining why it was sort of a challenge to pinpoint out the nodes, especially in lung cancer. Both also show some moderate centralization values, but it's closer to 0, meaning none of these networks have a perfect star-like structure dominated by a single hub. Maybe prostate cancer, but not that much. In this table, we can see how a high value in closeness centrality would mean that the nodes can efficiently connect to other parts of the network, while a low value in between centrality would indicate fewer bridging roles. So after analyzing the role of AKT in both cancers using Cytoscape and Venni, I was able to see significant differences in their structure, where prostate cancer is centralized and lung cancer is more distributed. The analysis revealed that prostate has greater network heterogeneity and centralization. These insights highlight AKT's role in both cancers, such as regulating cell survival and tumor progression, So in conclusion, AKT does play a role on cell survival in cancer pathways.